How can we do it? Ask Richard. You know who is Richard? Ask Richard. Yeah, she's or him. Welcome back. I hope that you enjoyed the lunch. Welcome back to the afternoon session. Our first speaker is Dr. Angela Maria Savino. She's a molecular oncology researcher, and she started her career here in University Milano Bicocca as a graduate student in translational and molecular medicine program of DMAT. Then she continued her research in Tel Aviv and then in New York. And her research is focused on novel therapies to treat Down syndrome patients with B-cell acute lymphoblastic leukemia. And now she's here back in Italy as a winner of the Rita Levi Montalcini Career Development Award, that is development award aimed to bring back to Italy brains that were spread in the rest of the world. And now she has a position as assistant professor at the University of Milano Bicocca, and she is a junior group leader at Fondazione Tettamanti.
Hi everybody, good afternoon. Thank you, Clara, for the nice introduction. I understand that I am invested now with a big responsibility. First of all, because it's the first talk after lunch, so I hope I can keep you awake. I hope you all had coffee so far. And also, after the great panelists of this morning, I feel very small. So I think my goal is just keeping you awake, so I will make an effort in doing so. So I'm going to present my research today, so what I've done during the past years, which is focused on targeting acute leukemias. And in doing so, I want to walk you through the journey, because I've been done this work moving a little bit around in different countries. So I have nothing to disclose, of course. So I'm going to walk you through this journey geographically. So I'm a PhD, so I'm not an MD PhD, so I really appreciate all the work you do, guys, but I'm a simple PhD. And I did my PhD in Milano, in University of Milano Bicocca. It's called DMET, it's a Translational and Molecular Program of a PhD program. And it's like 3 years program. And then for my first, after the PhD, I was very curious to explore new realities, explore new countries. And I moved for my first postdoc to Tel Aviv, where I spent another 3 years, and I was working, always worked on leukemia during all these experiences I've done. And it was a great experience for me, both culturally, because it was a very different culture compared to the Italian one, compared to the one I was exposed before. And also scientifically, because I was working with one of the best mentors I ever had in my whole career. Again, this topic of mentorship has been mentioned many times today in the talk. So I do endorse that this is a very important step in your career. Having a good mentor is very important. And I spent there 3 years for my first postdoc. I was studying leukemia and mechanism of Down syndrome-related leukemia. And after the 3 years, I moved. I wanted to explore even more. I wanted to do another different experience, and I moved to New York. I moved to Memorial Sloan Kettering. I wanted to do the American experience, so-called. And in Memorial Sloan Kettering, I worked with another scientist, less of a mentor, more as like a brilliant and a very ambitious guy. And we had a very nice relationship while I was there. I spent another 3 years in Memorial Sloan Kettering studying adult leukemia, in this case. So I moved to the adult leukemia. And the work we did together granted me the possibility to apply to this Rita Levi-Montalcini Career Development Award. As Clara mentioned, it's a program meant to bring the brain back to Italy, so people who have spent some time abroad can apply to this program. And it's a very prestigious one, and granted me the position as assistant professor and junior group leader back in Milano Bicocca. So I'm closing this circle, coming back where I started. And that's a very intense round that I did. So I've been traveling a lot, so I really felt I wanted to come home. But the main drive of all this journey, let's say, was science. So I wanted to really follow science. I really wanted to do better science from time to time. And I had a very specific thing in mind. So I was always focused on studying leukemias, so blood cancer. So I will give you a little bit of introduction of the normal development, of the normal process of hematopoiesis, which is normal development of cells, of blood cells, which is a sort of hierarchical process. And on top of this pyramid, you have the hematopoietic stem cell, which has the property of self-renew. So it's a totipotent cells that can originate all the cells of the blood, can self-renew to maintain this pool, and can differentiate in lymphoid cells and myeloid progenitors that can further differentiate in the mature cells of the blood, like B cells and T cells, and all the myeloid lineage that we have mentioned before in the previous talk. And this process is really finely regulated, so that's a fine-tuning upon the regulation of this differentiation process. So how do we go from the normal hematopoiesis to the disease state? So when some events can occur as in mutation, these events can occur at the level of stem cell or the level of progenitors, and this sort of mutation, a combination of events as in mutations, they cause a block in differentiation and an accumulation of undifferentiated myeloid progenitors in case of acute myeloid leukemia, or lymphoid progenitors in case of acute lymphoblastic leukemia. And these two diseases might be similar to some extent, but they are also very different. For instance, myeloid leukemia it's an adult disease mainly, and acute lymphoblastic leukemia, it's a childhood disease. And they're also different in pathogenesis. And although the cure rate for these two diseases is really high, so it's improved in the last years, still there is a high percentage of patients that relapse, especially in acute myeloid leukemia. It's really high, the percentage. So there is an urgent need to find new cures and new therapies for these patients to avoid this high rate of relapse. But in order to understand how to get a better cure, what is the main stage of the cure? So how do we actually treat leukemia now? So there are some historical treatments, including chemotherapy and radiotherapy, which is targeting general processes of hyperproliferation and survival of leukemic cells. And then more recently, targeted therapies, which are specifically indicated for some subgroup of patients having some specific lesions, immunotherapies and cell therapies, as you all know. And then ultimately, there is the stem cell transplantation. So there is a clearance of the sick cells from the blood that they are replaced by healthy cells from a donor that can replace all the normal blood formation. And in order to understand how to get a better therapy, to ameliorate this therapy, it's important to understand the mechanism of leukemia. So what are the challenges that the physician have when they treat leukemia, and what are the mechanisms they have to face? So leukemia, first of all, the leukemic cells lose the crosstalk between them. So there is a loss of cell crosstalk. So they become independent of the signals that come from the environment, as in cytokines. So they can start activating some pathways, some proliferation, some survival pathway by themselves without responding to anything, completely independently, which is very worrying because it's like uncontrolled system. Then they have a high energy demand. So to sustain their increased fitness, they have to rewire their metabolism in order to sustain all the processes that they are facing. And then they acquire the ability of invading other tissues. So they start in the bone marrow. Leukemia is I can't use the pointer, I don't know what is it. Anyway, so it's a bone marrow disease, and then it can invade secondary organs in the process called metastasis. So during my career, I've been explored these three areas of research, and I'm going to start my talk talking about signaling dysregulation, which is, again, the crosstalk of the cells, and how the signaling can be dysregulated and can drive leukemic process and development. So this part was done during my PhD. This is my group in Monza. I'm not even sure that I'm there, but anyway, I was part of this big group in Fondazione del Tamanti Monza. It's a place specialized for diagnosis and treatment of leukemia. So I started here studying acute lymphoblastic leukemia pediatric in children, and then I finished my this kind of studies in Israel when I moved to Sheba Medical Center for my first postdoc. So it was a mutual effort to address this topic of signaling dysregulation. Now, there are many signaling pathways that can be dysregulated in leukemia, and I've focused during all my career on JAK-STAT signaling cascade, which is one of the most important signaling cascade for proliferation and survival of the cells, and can be mediated by two biochemical complexes, receptors, and can be triggered by two different cytokines, IL-7 cytokines and TSLP. And this kind of signaling works in a way that gets activated by phosphorylation and activates through STAT5, eventually phosphorylation of STAT5. Here we are with the pointer. And it activates a transcriptional program leading to survival and proliferation. This is, again, the normal, how the signaling normally works for the cells. But there might, when the signaling is dysregulated, there might be some mutation in the receptor itself that makes the receptor independent of the signal, independent of the cytokine, and transcriptional activation and transcriptional control over the expression of one of the component of one of the receptors, for instance, this component CRLF2, which is overexpressed and triggers even more the signaling. So, and curiously, it's like this kind of dysregulations or mutation in the receptor and overexpression of CRLF2 are present in 7% of BLL, for instance, acute lymphoblastic leukemia, and causing an hyperactivation of this pathway. So I focused in the first work I'm going to present to you, I focus on the mutation of IL-7 receptor that can come alone, or coupled with CRLF2, with the expression of CRLF2, and these two mutations coupled together, they are very common in a subgroup of acute lymphoblastic leukemia called Philadelphia-like leukemia that, as you can see here, has a very poor prognosis. So there is a urgent need of finding more effective therapies for these patients. And also was interesting that both mutation of IL-7 and overexpression of CRLF2, they're very early in the leukemia, in the leukemia process. So they appear at diagnosis and then maintain at relapse. So we ask ourselves at that time, does this regulation of this axis, CRLF2 and IL-7 mutation, so triggering hyperactivation of JAK-STAT pathway, have a role in initiation of leukemia? Can this dysregulation prompt the leukemia to develop and arise? So to address this question, we did a very simple experiment. We overexpressed the mutation of IL-7 receptor alone or with overexpression of CRLF2 in precursor cells, C34 stem cells, human stem cells, and we transplanted these cells into immunocompromised mice, which is a general model that's used to model leukemia and to model blood cancer. And we analyzed the human cells isolated after engraftment, isolated from these mice, and we analyzed the development of B cells. So we had all the panel of B cell development, and we realized that there was an abnormal accumulation of early B progenitors in IL-7 receptor, here in red, mutated cells, and IL-7 receptor coupled with CRLF2 overexpression. So there was an accumulation of early cells that decrease in the maturation. So more accumulated, more progenitors and less mature B cells. So this was a defect. To get a better insight of this population, you have here a FACS plot. It's called showing the population of human cells that is being isolated from the mice. And this population shows the marker of early B cells, so it's CD19 and CD10, and within this big population, we identified a smaller population with the higher expression of this marker, and both of them showed the expression of a stem cell marker, CD34, which is abnormal in this case. Although we had this kind of abnormalities, these mice were not sick, so they didn't have leukemia, they didn't develop any kind of disease. So they had these human cells carrying the IL-7 mutation, but they had no leukemia. So what's going on? So we ask ourselves, so it's not causing leukemia, this mutation by itself is not responsible for the development of the disease. So we decide to give the cells another chance to accumulate disease, giving a stress signal. So in hematopoiesis, the transplantation, it's really stressing the cells out. So in the model, there is a primary transplant, and the cells can be subjected to multiple transplants. And when we did the secondary transplantation into the mice, we got full-blown leukemia. And this population, IL-7 receptor, this leukemia, didn't have only that mutation that we induced, but accumulated several other events, several other mutations. So it's like the presence of this mutation was predisposing the cells to accumulate further events and second hits, and to develop eventually the disease upon stress signals. So to better understand what kind of mutation the leukemia was showing at this point, we performed single-cell RNA sequencing from the population isolated from the primary mice, so what we called pre-leukemic population, this CD10 high, this CD10, CD19 population, and the leukemia that was isolated from the secondary mice, and the control cells, so cells that didn't have any mutation at all. So this, as you can see here, they cluster in space as a transcriptomic profile of the cells, so they have a very different transcriptomic profile. So the pre-leukemic cells cluster here, the leukemic are different, and the big population CD10, CD19 that we also define pre-leukemic, they are generally expressed. They are generally organized in space. So they have something in common with leukemia and something in common with the other population. So we were interested in understanding what specific mutation were characterizing, were differentiating different population. So among all the hits, all the mutation, all the alteration that we find, we were interested in this gene, CDKN2A, that was, as you can see here from the red dots, it was very much expressed in the pre-leukemic population. It was completely lost in leukemia. So that's very interesting because this is an oncosuppressor. So it makes sense that in the pre-leukemic population, the oncosuppressor was there, was holding the cells back, and it wasn't giving them the chance to transform. But when it's lost as a secondary mutation, then the leukemia is free to transform to full-blown disease. To verify this hypothesis, we did another experiment where we transduced cells with IL-7 receptor mutation, and we knocked out this CDKN2A gene, again in some CD34 positive stem cells, and we transplanted this in immunocompromised mice, and we got the leukemia at first place. So eventually the combination of these two mutations was inducing the disease. And so I can recapitulate this kind of model because we tried to model a model of disease in human. So this model tells us that IL-7 receptor mutation causes a block in differentiation in the maturation of pre-B cell, creating a sort of pre-leukemic population that is predisposed to develop the disease, but then it needs another mutation, another hit, another sort of alteration to actually develop the full-blown disease. So it was the first time that a pre-leukemic population identified with this IL-7 mutation was actually described in human cells, because in the mouse it's been described extensively the role of IL-7 mutation, but in human was not really clear before. So now, in the second part of my talk, I'm going to focus on the metabolism of cells. So dysregulation, always the key word is dysregulation of normal processes, so dysregulation of metabolism of AML. So this part was carried out in New York when I moved to Memorial Sloan Kettering, and this is my lab in New York. That was Michael Karas, my boss, and that was me, if you didn't see it before. And then you can see here like beautiful and stunning views of New York City. This is the view from my lab. That's the view from my house where I was living in New York City. And that's the celebration for the 9/11 memorial. So that's a very intense moment the New York City people has to live. So where two beams of light are projected instead of where the Twin Towers were. So it's a very intense chapter of my life. And so why studying metabolism of leukemia? What is the purpose of studying this kind of aspect of leukemia? So first of all, because the microenvironmental factors are always more and more studies and more known to modify the fitness of leukemic cells. So there is a mutual both sides effect of modifying, of environment modifying the leukemia and vice versa, leukemia modifying the environment. So it's growing interest in understanding this kind of crosstalk. And lately, also mutation in genes involved in metabolic pathway, as you can see, a mutation in mitochondrial genes, they are found their mutation in leukemia. So they must be a role, they must have a role in the development of the disease. And secondly, because, and finally, because also many drugs targeting enzymes, so targeting metabolic players, are recently approved by FDA, by EMA. So there is a growth. So all this kind of information gives us the idea that metabolism of leukemia, it's a worthwhile area investigating. So I'm going to summarize here very briefly. I'm not going to go into details, but the main sources of energy for the cells are glycolysis on one end that uses glucose as main source of energy, shunting through this pathway and produces lactate. And this pathway can happen without oxygen, so anaerobic condition. And then cellular respiration that happens only when oxygen is there. So those are the two main sources of energy. And as we said before, in this case, I was studying acute myeloid leukemia, which is a bone marrow disease. And the hyperproliferation of the cells in the bone marrow and they're crowding, they're filling the bone marrow. I'm sorry. And these cells are highly glycolytic, so they're using a lot the nutrients in the bone marrow, and they're also modifying the environment. So the hyperproliferating cells are creating dysfunctional vasculature, so it's more difficult to bring oxygen into the place. So we have a hypoxic environment eventually, and also they're consuming a lot of nutrients. So there is a shortage of nutrients eventually in the bone marrow niche, especially glucose, which is the main source of nutrients for this patient, for the cells in general. So in this condition, so it's sort of it becomes a sort of a hostile environment for the leukemic cells and for the cells in general. So what happens, how do the leukemic cells survive in this environment when there is no glucose, there is no oxygen? What do? They need to exploit different sources, alternative sources of nutrients in order to survive. So how about fructose? Fructose is very similar structurally to glucose, so they have a very similar structure and very similar molecules, and it's very abundant in Western diet. Taking into account that this study was done in US, so in US it's very prominent, the presence of fructose in the diet as by itself, like fructose as single molecule, especially this high fructose corn syrup, which is very abundant in the diet, and also in a complex form of sucrose, where you have half of the molecule, like the molecule of sucrose is made half of glucose and half of fructose. So the fructose is there and gets consumed. And also it's less sweet than glucose, and it's less palatable, so it prompts the people to consume more and more. And it can lead, this high consuming of fructose can lead to, to obesity, for instance, and obesity, for instance, is one of the risk factors to develop leukemia, acute myeloid leukemia. So it was really interesting for me to understand how can this fructose can contribute to the development of the disease. And also, there was a study showing that there was an abnormal accumulation of fructose in the bone marrow of AML patient compared to the normal patient. So fructose is there, is available. What do the cells do with it? Can they use it? So the question was, can leukemic cells exploit fructose as source of energy instead of glucose when glucose is missing? So we did a very simple experiment to verify this. And we grown a panel of AML cell lines. Those are all AML cell lines, more or less. And they were grown in glucose and fructose. So, and as you can see here from the proliferation curve, they have a similar growth. So they can use fructose. They use it fine. They can grow fine in presence of fructose. But they show a very different metabolic program. So we performed some nuclear magnetic resonance analysis to quantify what kind of metabolites do we have in cells when they're grown in AML cells, when they're grown in fructose and they're grown in glucose. And in fructose compared to glucose, they tended to accumulate these two metabolites, glycine and serine, and to downregulate this lactate, which is the downstream product, as I showed you before, of glycolysis. So they have less glycolysis. Here it's a bit counterintuitive, but the fold change is negative, so this is going down. And that's and they are accumulating these metabolites, glycine and serine. So what do they do with these metabolites? We ask ourselves, what is the purpose of accumulating these metabolites? So basically, they have two chances. Like the cells, they face fructose. When fructose is there in the environment, they can intake fructose. They can either shunt it into glycolysis or break glycolysis in through and activate the serine synthesis pathway through accumulation of serine and glycine. Interestingly, this accumulation of these two metabolites, as you can see here, they have more serine and more glycine in fructose, happen only in the leukemic cells. It doesn't happen, it's not significantly happen, in the normal hematopoietic stem cells. So the way they use fructose is typical of the disease and is sparing the normal cells. So that's a very important distinguish. It's very important to distinguish these two pathways. So it's selective for the leukemic cells. And so, but how can we understand if this upregulation of the serine synthesis pathway makes the cells more dependent on this pathway? Because if the cells are dependent, we can target this pathway and we can make the cells more vulnerable. So we were creating a sort of vulnerability. So in order to understand the dependency, we inhibited the PHDH, which is the first rate-limiting enzyme of this pathway, and then we ablated the production of serine and glycine. So what happened when you block this pathway? Are the cells really vulnerable to this blockage? So as you can see here, we used a system to knock down PHDH, this enzyme, which is called shRNA. It's a system to knock down, and as you can see here from the Western blot, you don't have PHDH anymore. And upon knockdown of PHDH, the cells were not doing glycine anymore. So this is the peak of glycine that you can see in NMR spectra. Less interesting, but anyway. So they don't have this peak anymore. So they were not doing glycine anymore when you knock down PHDH, and also the viability of the cells in fructose was decreased. Particularly in fructose was decreased when you knock down PHDH. So the cells don't have PHDH, they cannot use fructose, they die. They proliferate less, let's say. And also they responded to the PHDH inhibitor in fructose, as you can see here, the proliferation in a dose-dependent manner, and the CD34 normal cells didn't respond at all. So again, specificity versus towards leukemia. And in vivo as well, we transplanted cells, AML cell lines, carrying the knockdown of PHDH. So the control cells, they have PHDH, and the knockdown, they reduce the expression of this. And we treated the mice with glucose and fructose, and in fructose there was even in vivo, there was a huge decrease of engraftment of the cells without PHDH, as well as treating patient samples transplanted into mice. It's called PDX model, patient-derived xenograft, and treating this these mice with the PHDH inhibitor, so the drug was blocking PHDH, caused a decrease, a huge decrease in proliferation, in engraftment of these cells upon treatment with fructose. So eventually, we identified a new metabolic vulnerability in AML, which can be triggered by the presence of fructose there. The serine synthesis pathway plays a critical role in the metabolic adaptation of the cells in the setting of elevated fructose, so always when the fructose is there, and the PHDH inhibition can be a promising therapeutic approach promising an alternative therapeutic approach for the patient with AML. For the last part of my talk now, I'm gonna show you a work we did on metastasis, and specifically metastasis into the central nervous system. So for this part of my talk, we are back to in Israel, because my interest in metabolism actually started in Israel when I was at the end of my second postdoc before I moved to US. And I was part of this. I was living in this beautiful country for 3.5 years. Of course, the country has its own issue, but you can have, the sea, you can have the mountains, and then you can have the desert. There's anything. They claim that they have everything they wanted. The only thing missing is really peace, but this is a long story. So I hope that someday this problem will be solved. Anyway, I was there to do science, so I explored metabolic adaptation of ALL, so acute lymphoblastic leukemia, to the central nervous system. And when the leukemic cells can invade the central nervous system, but they do not invade the parenchyma, so the gray matter, but they stay into this interstitial space. It's called subarachnoid space between the meninges. So they live, proliferate, and survive there. And they are bathed by the CSF, cerebrospinal fluid, which is the main source of nutrients for these cells. So nowadays, every child who gets diagnosed with leukemia, independently of the involvement of the CNS, gets prophylactic intrathecal chemotherapy. And as you may know, by the time it reduced the CNS relapse rate, although to around 5%, I think, but the median overall survival is still very poor. So although they relapse less, but the people who relapse, they are doing very poorly. And also in the long run, you can have it can cause secondary brain tumors and neurocognitive defect because it's a very invasive procedure. So there is an urgent need, again, to find alternatives for this kind of therapy. So since I was interested in metabolism, I kept going with this question, and I asked myself if the leukemic cells can display metabolic vulnerabilities in the CNS. And to answer this question, I did again another experiment, pretty simple conceptually, where we injected ALL cell lines into the mice, and we isolated human cells from the brain and from the spleen, but it might as well be the bone marrow, so from the primary site and the metastatic site, and we performed RNA sequencing on these cells. And from the sequencing, we gathered that there was an upregulation in the brain, in the cells isolated from the brain, of fatty acid synthesis. So genes belonging to the fatty acid synthesis pathway were upregulated, and there was a downregulation of the fatty acid degradation. So it's like they were building biomass, and they were less on the energetic side, like gathering less energy in this case. So we ask ourselves, why are they synthesizing more fatty acid in the brain? What's the composition of the brain? Why do they need more fatty acid there? So we explored the composition of the brain, and as you can see here in light gray, that's the CSF amount of fatty acids in general, and it's way lower than the bone marrow, than the plasma, for instance. So the cerebrospinal fluid was very poor in fatty acids. So the cells get there, they need to adapt, and they start building their own fatty acid because they don't find it in the environment. And one of the genes that was particularly upregulated among the signature of the fatty acid, the lipid synthesis, was SCD, steroid-CoA desaturase A, and it's involved in the maturation of the fatty acids. So in order to have mature fatty acid, to do all the processes that the cell need them for, membrane phospholipids, energy storage, and lipid signals, they have to be mature. And this SCD is responsible for this maturation. So we measured the expression of SCD in patient with the bone marrow diagnosis. They were diagnosed at bone marrow, they relapsed at bone marrow, and they relapsed in the CNS. And as you can see here, the expression of SCD was way higher in the patient that relapsed in the CNS. So this is an important target. That's something that we want to pursue because it's really much upregulated. So we modeled this. So in the lab, we overexpressed SCD in cell lines, ALL cell lines that didn't have higher expression of SCD. As you can see here, there was a difference So this is our control cells, and that's SCD high cells. So we were gonna call the cells SCD high. We transplanted this SCD high into NSG mice here, and then we analyzed the engraftment, so how many human cells we find in different compartments of the mice. And as you can see here, there was an accumulation, a fast accumulation of cells overexpressing SCD in the CNS, and this didn't happen in the other compartments. So this effect was specific for the CNS. And when we inhibited SCD, on the other hand, we transplanted, first of all, cells from patients into immunodeficient mice and used an inhibitor, SCD inhibitor, and as you can see, there is a decrease in CNS engraftment. They didn't affect the bone marrow. So again, a very strong effect on the CNS. So at the end of the day, we can conclude that the leukemic adaptation also to the CNS is metabolic, it's primarily metabolic. So there is an upregulation of de novo fatty acid synthesis, which is crucial for the adaptation because the environment of the CNS is deprived by the lipids, and the overexpression of SCD leads to a clear phenotype of earlier CNS involvement in vivo, and the SCD could be a promising target a promising target to cure these diseases as alternative therapies. And so to conclude this, so we analyze two different cell lines. So we analyze, two different leukemias, AML and ALL, two different compartments, bone marrow and the CNS, two different metabolism, sugar metabolism as in fructose, and lipid metabolism, and we identified two targets. So that's some sort of crosstalk between basic science and translational science, because eventually, at the end of the day, we identify these two targets that can be effectively therapeutically targeted to get better, get a better therapy, as we say. So my ongoing research program, so what I'm planning on doing here in University of Milano Bicocca, I want to investigate the non-metabolic genes, mutation in these non-metabolic genes in leukemia, and what is their role to induce new metabolic vulnerability. What can the diet modulation, how can the diet modulation affect tumor progression in this case, leukemia? So how can we play with the diet in order to restrain the tumor or push the tumor or increase the effectiveness of the therapy? And then I would like also to investigate the role of metabolic players in leukemic stem cells, so in the cell of origin. And this is how I think about organizing my lab. I would like that it will be like a mixture of scientific skills and soft skills, and also humankind. So I would like to combine like the availability of patient samples, so studying some sort of mechanism on patient samples, using preclinical models as well, a mixture of molecular biology and metabolomics. But also it's very important to have team working, like an effective team working in my team, creative thinking all the time, and the networking. And then networking was also mentioned many times during the talk, and I think it's very crucial for the development of the scientists and the science community. And with that, I conclude, and I thank you for your attention, and I would like to thank all the sponsors and the places where I've been around the world.
Thank you, Angela. For a matter of time, we will make our question to you later on, okay? I have a question for you, but later.
So, are you wondering if a parallel career in science and surgery is possible? Our next speaker is a great example that this is something attainable. It is a great pleasure and honor for me to introduce you Professor Pascal Senn, Chairman of the Service of Otorhinolaryngology and Head of the Inner Ear and Olfaction Laboratory in Geneva, Switzerland. Professor Pascal Senn devotes his clinical time to treating and to helping patients with hearing loss regain their hearing function, and in the meanwhile, he explores new therapeutics for hearing restoration and autoprotection in his lab. I hope that his talk will motivate and inspire you as he motivated me to get involved in research. Please welcome Professor Pascal Senn.
So, dear Dimitrios, dear organizers, thank you very much. It's a great pleasure to be here. And I was listening to Professor Citerio this morning, and actually he said everything I wanted to say. So luckily, I actually not following the program as much as I put it here into the title. So actually, I will tell you something about my clinical activity, followed by actually my research activity, and then how I combine both. This is my field of work and science. It's the ear and the human ear that actually we want to help treat it in the future better than today. So, because in our beautiful world, there are over 1 billion individuals with hearing loss, actually, and of which 500 million has invalidating hearing loss. That means they have actually consequences, negative consequences on their educational, professional, or family evolution. They really suffer from that in a really important way. And for your general culture, we have 2 kinds of hearing loss, 2 main types, of which the first one is a mechanical. It's a transmission hearing loss. It's actually located where the problem is located in the external ear, like a clock, or the middle ear, that will block the sound waves of coming to the eardrum. And we have the sensory neural hearing loss that is actually more related to the inner ear or the hearing nerve. So the first is here, as you see here, the eardrum. If the eardrum, it's beautiful if you see it here. It's really, and the name, I was really fascinated. It looks really like a drum, and it's in the ear. So the terms are very intuitive and logical, and have the same functions. They vibrate. So you can imagine if there is a mechanical stress, a rupture or something on it, you will hear less, and you will hear differently. And that's actually my field of clinical work. We just repair. In this case, we repair the eardrum. And we do that by using tissues from your muscle. The white parts of the muscle you go by in the butcher's place, the white he cuts away because you don't eat it. It's perfect to repair actually the eardrum. It's the fascia. This one can be used. And you see it here. We put it under the remnants of the eardrum. We support it from below. We can also change the ossicular chain, like you see it here. Like in Geneva, we have a long tradition of watchmaking. Also, I use the same microscope as Rolex, actually in another edition. But it's very beautiful to have it here, and I must say they inspire me as well, this kind of microsurgery. For instance, if we have the smallest bone in the body, it's called the stapes, and you'd see it here. It's one of the 3 ossicles that makes actually the link between the other ossicular chain and the inner ear. And if this one gets blocked, we have to remove it and replace it. As you see here, again, through the external ear canal with microinstruments under microscopic view. Here we lift it off the eardrum. We see here the stapes. With a laser, we cut it off. We make a hole into the inner ear, and we put the small prosthesis, which has 4.5 millimeter length and 0.5 millimeter width, so it's very small. Don't drink before surgery. And I show you just a brief extraction. This is the cut in the external ear canal. Here you see the ossicular chain with the blocked stapes. Here I drill, and then I measure, and then I put the prosthesis. Now you please check the size of my finger compared to the prosthesis, and we place it there. And we will then connect it. And now the movements are transmitted into the inner ear again. And what happens here is that the hearing threshold that was down here gets up here, so almost a normal hearing. This is a mechanical kind of repair, which is beautiful and elegant and takes me about an hour. Now the inner ear is different. The inner ear, as you see here from a friend of mine from Uppsala, Helge Rask-Andersen, who made an electron microscope view of the inner ear. You see here the tunnels, with here the organ of Corti, and here is a magnification that he colored. You see here the outer hair cell rows and the inner hair cell, and here the auditory neurons. If these cells are going to be damaged, they're not actually repairable today. So, for instance, this means that loss of inner ear hair cells leads to irreversible hearing loss in mammals and in humans. As you see, noise, disease, aging, genetic deficiency, infection, trauma, drugs, and many things else, they will destroy these hair cells and leave just a terrain of burned soil, and it's not repairable so far. So the treatment for that are hearing aids, as you may have seen by family members or in the bus sitting behind an elderly person. And for those who have severe forms of hearing loss, really severe forms, that hearing aids are no more sufficient, we put cochlear implants. Now, hearing aids that you have seen before work with amplification, but cochlear implants are actually neural stimulators. They're inserted deep into the bone here, and then the electrode wire is inserted into the cochlea where it stimulates directly the auditory nerve. So it's a neural stimulator. And this is compatible with complete hearing loss. So people who cannot hear anything anymore, 1 year in general, 1 year after using this device, they're able to telephone with people that they cannot, that they don't know. So it really restores hearing to a functional level. It's actually a miracle of biotechnological engineering. So what my patients, they ask me, do I have to wear a device that is this big, this actually stigmatizing? Is there not something else? And they always ask that, and that's why the research field actually in which I'm working in, we are looking to find a better therapy for hearing loss in the future, which is more natural, which is more efficient, which is invisible, durable. Like, for example, something like the hearing repair that I showed you before. They really dream of that I can do a surgery that lasts 1 hour and they would hear back again. They talk about that. Why can't you do it to me? You did it to my uncle. And I said, yeah, but he had another problem. Or a medicament they could take that would reverse it. And here is the champion, like the birds, the chicken. Because they found out in 1988, actually 2 different groups independently, they wanted to do experiments with birds, chicks, that they blasted with sounds until they were deaf. And then they were waiting to treat them. But when they get to the experiment, they found out they were hearing again, completely. And so, independently, the 2 groups, like many discoveries in science, were done independently in different labs. This was the same. You see here, this is the basilar papilla of a bird. After the blast, with the sound, they were losing hair cells. But it came back over time. After 2 weeks, it was perfectly restored, and they could do it over and over and over. So this discovery made really a turning point. Many researchers and patients started to dream about that. How can we apply these mechanisms for our human therapy? But just this is my student that loved micrography. And he actually, this is now in green, you see hair cells of a mouse, and in red, the auditory neurons stained with TUJ. And he did it with, he made the cochlea translucent, and then he put antibodies on it. But you see the complexity is different. It's a three-dimensional coil over 2.5 turns to repair this is not as simple as the carpet that the bird has. So the exact anatomical position of the cells is much more critical. But the biological therapy of hearing loss with regeneration of lost cell types, this is a dream. And we actually started in 2003. This paper came out by Heller in this group, Huawei Li, where they were showing that they could remove the inner ear from mice, put the small organs into a single cell solution, growing neurospheres, and could produce hair cells in vitro and in vivo, so that it was possible to transfer the principle in limited way to mammals. And actually, this was my starting point, because I wanted to do that in humans. And being able to do surgery, having access to human tissues, it was actually my goal to reproduce it. And so what you see here is the temporal bone of a human body donor that we then actually worked on here. This is the eardrum. We removed the inner ear organs. Here is the ampulla of the vestibular system, the utricle of the vestibular system, and the cochlea. I show you now just some small films, if it works. Yeah, you see how we drill here the ear canal to have more access to the eardrum. And then here we would remove the stapes again. I'll come back to it later, why I show you that. First of all, because it's beautiful, but there's another reason, I'll come back to it. But you see here, the stapes is removed. We see here the utricle. And here is, after enlarging, I will remove the utricle to put it into culture. Again, I'll come back to it, why I show you that. You can live without it, I know. But what we did scientifically, we would then put this utricle, we would mechanically dissociate it and put it into single cell culture, give growth factors for several days, induce neurosphere formation, cell separation. Again, here was actually a loop of amplification, or we could plate it at any moment to do immunocytochemistry. And you see here the sphere, the neurosphere, which is actually a stem cell surrounded by its daughters, so floating. And actually it was possible from humans to remove it, especially from the utricle, but very rarely from the cochlear duct. Everybody was talking about hearing loss, so utricle is for balance. So it was much more possible there than for hearing loss, which was a little bit disappointing. But we were still able to grow inner ear hair cells from human origin that we could use in our lab. Although it was particularly difficult to publish it, it came only out in 2020, although I started in 2005. Because human cells grow slowly, because it was so many obstacles with ethical licenses, whatever. But it was about the topic that I will show you later on about resilience and stand up when it doesn't fly. It was really slow, but it worked. And in the meantime, we received so many grants for this specific question because we're actually the only ones having access to human cell types of the inner ear, which gave us a huge advantage to work with that. And with my postdoc, an Italian, really brilliant lady, Marta, we were able to refine the protocols for human fetal hair cells. We could then really produce it in higher quantities and select subpopulations. And actually, again, was a paper that was covering an area that nobody else was working in mainly at that time. And now I'm working in Geneva in my lab, and I had another guy, Francis Rousset, which I showed. I came from another university of how I did it with the mice and the humans to extract stem cells and to put them in culture. I showed him the first time. He didn't get back to me until 3 months later, telling me, what do I do with the freezer full of cells? And I said, nobody went beyond the 3rd generation, don't fool with me. He by chance found a cell line, a mouse line that has indefinite propagation of auditory neuroprogenitors by chance. So he could go into passage forward, he could freeze them, thaw them, just by chance he found it. You see here, this was a normal black 7 mouse, a black 6 mouse, and this is the passages he made. But the good news was that he would actually start to investigate what was the difference. He made an RNA-seq, compared different mouse types, and he found actually 2 pathways, Wnt agonists and SMAD inhibitors together, and he could do it with any other mouse now. And we are now about to try it in humans. And it seems to work as well. I'm not talking about hair cells, I'm talking only about auditory neurons. But the good news is really, with this, we have now a protocol to make human auditory neurons without limit, and we can use them for physiological experiments without limit when we need. And his last part is actually now we introduce fluorescent reporter genes, M-cherry for a neuron and GFAP in green for supporting cells. And now this one is completely animal-free, meaning as soon as from this endlessly propagating mouse auditory neuroprogenitors, we differentiate neurons, they will light up in red. And we can immediately also in another analyzer make a glutamate test from the calcium release and have a functional analysis also. We don't need antibodies of animal origin. We don't need any more new mice. We can all do it based on that. And this actually gave us even, we got the prize from the animal advocates. You have first to get that if you do animal research, that they give you a prize. I'm really happy because auditory neurons, they're very, very rare. We don't have very few. And if you have to kill many mice to have this kind of cells, but with this amplification, we can produce endless, and it costs less, it's animal friendly, and it's really reproducible. So replace, reduce, refine. Your generation will be facing a lot of these kind of questions to do animal research. Do you have to feel bad if you eat ossobuco milanese? I don't know, but it's a question today you have to face. If you do animal research, if you can propose something that replaces it, they will be happy. You will get other grants and you will get promotions for your young people also working in that field. So the inner ear regeneration is still far from being a therapy, but we have found the auditory neurons. So I put this slide so the chick, they don't have to be afraid of us because they will still be ruling, and you shouldn't look them in the eye directly. It's just not to provoke them. That's what I learned. Hair cell regeneration remains a challenging. So I've talked about the neurons. For the hair cells, we have to continue, which we do, of course. And what we learned can be used for others. Just a brief example. What we learned about the auditory neurons, me as a surgeon, we can have this kind of research for applied science. So the inner ear has 3,500 individual channels, like it's perfectly fitting the TV, which has 4,000 channels, 4,000 pixels, right? The cochlear implant has 12 individual channels, and it still works. Imagine the compression on your TV if you go from 4,000 pixels to 12. Who would buy a TV like that? But people can hear. But it would be great to improve it. Why do we have so few channels? If you see here, there is a distance between the nerve in green and the electrode in the inner ear. And if there is the electrode firing here, it would all, due to the distance, stimulate groups of cells at the same time, hundreds of cells actually. It would be like if you have a key of pianos, but you would play it with gloves. So you don't hit one single note, but you would hit 100 at the time. Maybe you can listen to Happy Birthday somewhat, but Rachmaninoff will be difficult. So what we wanted to do is actually to bring the nerve down here. And to this, we had to fill the cochlea with a matrix, because the neurons are non-swimmers, they don't go into water, so the inner ear is filled with water. We had to provide them with a scaffold, and we had to change the surface of the electrode by nanotechnology, by factors that are released. And actually, it was me as a young clinician-scientist who could direct an EU program based around that, where we were 4 university hospitals, large industry, 2 SMEs, 2 applied universities to try to tackle that. Because I had access to these human tissues, we would get the grant, and as a clinician-scientist, I understood the challenges in science and also on the medical side. And we were actually able to see, this is a deafened guinea pig, you can see that, oops, sorry, jumping here. You can see here that the neurons, they really came down and they were contacting now. We were able to bridge the gap, meaning in theory, we could have now much more channels because we need less energy and we are more selective. So this is what we learned also. So I talk about the combination. So this was my boss, who actually, Rudolf Häusler, who was in Bern, who actually preferred to be with us than with his peers at the conferences. He would hang out with us or take us on his boat. But he said, if you are going to be away from surgery for 2 years, you will lose your surgical skills. Because I told him I want to do regeneration, I want to go into the lab. He said, forget it, it makes no sense. He was very charismatic, but he was wrong. But he could take it. He would say, if you still want to do it, okay, then do it. And he was interested in what I do. He didn't believe. But he let me do, and he came even to visit me in Boston when I was there. And you see this here, all that is actually surgery, right? I wouldn't have had access to these tissues. Who has seen a cochlea of human? This is the organ of Corti with the neurons attached to it. Who has seen it like that? Which surgeon goes that deep? I could do it in the lab, but I wasn't aware. I was only aware afterwards when I did surgery again that I had an advantage over my colleagues because I had access to it, not the others. And I told him, he said, you were right, but still you shouldn't have done that. So what it takes, and it has been said, and I don't have to repeat it, I think what is really the most important points are here, and what it offers, as I said, insight. I think it's the insight. It offers you insight, and you go beyond what has been done before. Surgical training, what we have done in the humans, I did it in mice. The newborn mice, we took out the inner ear, we took the epithelial sheet. It's much more complicated. So when I came back after 2 years of exclusive research, I was quite shy. I thought, I won't be able to do surgery. I was away, boss told me. And they sat me there before the stapes. Many colleagues, they said, now let's see what he does. But for me, it was easy because it was much bigger. It was for me a hole like that compared to the mouse inner ear. And I had a perfect control. I wasn't expecting that. I was giving me a chance to not to be ashamed, but I was giving me a chance. But it worked out brilliantly. So only afterwards I started to tell you, doing a fellowship in research, an MD PhD in a surgical field where you do animal experiments or cellular experiments manually, you train your eye and your hand coordination to a level that is beyond what is needed in surgery with humans. Nobody told me that. I've never heard it before. So I tell you. Remember that. And the true link between science and clinics, when I was hearing your talk, I mean, who will understand the new immune therapies? Who will understand the new therapies that come into clinics? It will be you if you have some kind of really dedicated focused training before. You will be the ones that will translate that from the science to the patients. And it is badly needed because therapies will become so complex now that we have difficulty to understand. But you have a chance to go beyond to understand it better. And you have answers to your patients' questions, and you will be more independent because you know how to overcome problems that the projects you wanted to do are not running. You know, keep on kicking, go ahead. So for me, to become a relevant scientist and a better physician for your patients, all of that is what it offers, really. You do what matters. You will do what is interesting, but you will also do what is relevant for the patient, which was my starting point. You can't imagine how many of my patients that I operate every day are talking to me about cellular therapy, genetic therapy, how much they want to know about it. At least I can tell them what I know, but it's a little bit more than what my colleague knows who never did this kind of research. So they really want that. They want you to be translator for that because they read it in the newspaper. So with that, I want to thank my lab, and you see Dimitrios is already here. And I tried to pair, as a head of a department, a basic scientists up here to connect them each with a pair of a doctor so that they are really together in one project. Cisplatin ototoxicity is one. It's him together with German. Then Dimitrios will work on radio-induced ototoxicity. He will work with Francis. We have also olfactory regeneration, where we have Basil and Julien together with Rebecca. Vivian is especially in regeneration as well. So I try to build pairs, but we're very small, and this is great to be the boss. You can do whatever you like, and I like to be with the young people and let them play. Because spare time, it's quality time for you to do research. So go ahead, please enjoy whatever you can. We have nice collaborations all over the world. We have financial support, and with this image from Geneva, I would like to thank you for your patience. Thank you.
How can I activate? Thank you, Professor Senn, for this fascinating and really inspiring presentation. Are we have any questions from the audience? Someone who would like to do surgery and combine it with science, maybe? Yes, Maxime? Just wait.
Yeah, okay. Hello, yeah, hi. I have a question regarding the slide that you described regarding the cochlear implants and how coarse the ability we had to excite individual nerve cells was. Have we ever recorded nerve cells while stimulating them with this cochlear implant to know what the ground truth is regarding how many neurons we stimulate at the same time? Is it possible, especially in your ex vivo preps?
Yeah, we did that. It was, as you've seen, an international collaboration that was done in Tübingen. We did it in guinea pigs, and we could see that guinea pig electrodes, cochlear implants, if they had these nanotechnological modified ones that had gapless interface, they had impedances that were much better, they used much less energy, they were much more responsive, so the concept was right. We could see that, yes. But we couldn't ask the guinea pig how much it heard, really. But they heard better, that's for sure.
So you're recording single neurons?
No, at that time, no. We did this kind of compound action potentials. We were reading from the nerve, but we could see how much energy we would have to put in to get an answer, and we could see that we could reduce a lot the energy needed to go in. So impedances we could test, and actually, this kind of neural responsiveness.
Okay, and you also did that with the new type of electrode where the nerve is now in the matrix that you inject. Okay.
Yeah. Now it is also the frustrating part. Now it's up to the companies whether they want to go down this road or not, because cochlear implants work so nice today. Do we take the risk? Don't we? We'll see. But at least we could prove that it's possible in general. But then we do something else.
Thank you.
Thank you.
You can also put questions for the first speaker. We grouped the questions, okay? So if you have any other questions about leukemia or the research, you can address at this moment.
Thank you. May I ask very generally, as a surgeon and a researcher, how do you split your time?
Yeah. Thank you for the question. Actually, I'm also head of the department. So I do, I don't know how I do it, but what the good thing is to have a team everywhere. I have excellent colleagues in the lab and in the clinics, but overall I operate on 2 days, I consult on 2 days, and I do 1 day for research, but management of the service as well. So sometimes it's also in the evening or so. But the good thing is, and I think I learned that from a professional musician. He told me once, you know, the ones who play in the famous orchestras in the end. I mean, there's so many good musicians. Also, to have a paid position in a good orchestra, what it needs is that you keep on having so much fun and to take out more energy than you invest, despite all the endurance and the hardship you have. And this is a good thing about research. If I go there, I'm young again. And Dimitrios can prove it. I would not play the card of being a boss at all. I really go there. We're all friends. We go drinking. I learned that from my boss. We went sailing together. We have no hierarchy in the lab, really not. And this is, for me, it's going back to school with my buddies. Although I'm technically, I would be their boss, I never play that card, especially not in the lab, because I can feel young again. So it keeps me young. And this, I would say, is actually one maybe of the keys why you intuitively you would do it. You will keep young if you stay in science. But I'm not saying, don't ask my wife this question.
I actually have a question for the first talk. I was wondering, you said you worked with both children and adults, so I was wondering, talking about the metabolic dysregulation, what main differences are between if there are children and adults, and how that plays a role in the context of the SED inhibitors and CNS intervention you touched upon in the end.
Thank you for the question. Actually, it's a very good point, because the metabolism it changes during the evolution, during the growth of the people. So we assume that there are different metabolic requirements according to the age. So there might be also different metabolic diet requirements if you are targeting different ages. So for sure, in the lab, like when you work with that's one of the problems that we have as researchers. When you work with models, you can't really perceive that. It's more on the person, like later on when the research gets translated. But for sure, you have to consider that the development plays a role, and the requirements, the nutritional requirements plays a role when you consider the impact of the therapy that you're also gonna use. For instance, so for this SCD that I mentioned at the end, we are planning on, so this is not clinically graded, of course, but we're planning on studying what kind of impact can I have on the children, and it will be mainly a childhood therapy. So this is under investigation. So we're just trying to figure out, I mean, whenever it will be promoted to the clinic, figure out how the doses, everything changes between, but this is also in general, for metabolism specifically, because the requirements are really different. So that should be calibrated according to the patients that we are trying to target, yes.
Any other questions?
I also have a question for Dr. Savino. I was wondering, other than winning the Rita Levi-Montalcini Award, is there any other reason why doing research in Italy is appealing?
Let me think about it for 5 minutes. Actually, I mean, we have a drive. I mean, all the Italians that go abroad, I think, I'm not sure about that, but you can always feel a drive to come back to your country. So like, it might be less appealing research-wise, but there is also a personal drive that is, driving people to come back. That said, I think that of course there are differences in means and availability of funds, and the way you can do your research. Especially there is a difference in time, because everything in Italy, that's my impression when I came back, it's a little bit stretched, but this does not mean that it's of less quality, because you can do, I mean, there are brilliant scientists in Italy, you just need to work a little bit more on the connection, it's not so fast. That's my impression. But you clearly can work, again, on the connection. Beautiful people, brilliant people, very smart, and they can interact in some sort of networking that can really help the research. So I think that's one of the most appealing things, that you can come back, you have like a flourishing community that needs a little bit of a push, I think, to move things forward. But we have a huge ability of understanding science, and also our preparation, our degrees, they're really strong, because I think like theoretically we are very well prepared. So all this kind of knowledge can be exploited, and it has to be worked out a little bit more, but it's, I mean, it has a great potential. Let's put it this way, yes.
A very last question before we move to students' presentations? Or none at all, because we're already 5 minutes late. Thank you so much to both of you. It was very inspirational.